hush falls over the crowd. Hi, Jeremy. I'm ready. Well, the way it'll work, I don't know if Sam explained it, there'll be, you've got 15 minutes, but we won't have some time for questions. So there's 11 minutes of green and then 4 minutes of yellow. If we can have everybody take their seats, that would be wonderful. Or not, okay. All right. Thank you all very much for coming back for the afternoon session. We've seen a lot so far today in terms of information on the environment and on the animals, the effects of the environment on the animals. We've seen a lot of information about how the environment is changing and how our needs for information about these animals are changing. What we're going to see in this next session is some of the ways that we're being able to address some of these problems and issues through emerging tools and technologies. So we'll have a series of speakers talking about telemetry, about sail drones, about passive acoustic monitoring, and environmental DNA. Our first speaker for this afternoon will be Melinda Holland with Wildlife Computers to talk with us about advances in telemetry and biologging instruments.
Thank you and good afternoon, everybody. I'd like to again thank this group for the opportunity to share these advances in telemetry. Wildlife Computers has been making electronic tags for marine animal studies for 31 years. Some people in this room use some of the very first tags that Roger Hill developed. I've been lucky to be a part of this company for the last 28 years and help support the growth in the use of electronic tags from being addressed to just a few select researchers to becoming a globally used tool. During this time, the use of electronic tags has grown from being a cutting-edge, high-risk technology that started recording how deep an animal could dive. Now it is considered a standard item in the researcher's toolkit. But as the questions have become more complex, fortunately, there has been a concurrent advancement in the technology in general. Does everybody remember your very first cell phone compared to what they are now? Tags, like cell phones, have become smaller and smarter. So I'm kind of going to go through the different parts of the advancements in the technology, and one is the sensors and the types of data collections, and the questions these answer. So one of the very first questions that has been asked is, where is my animal? And marine mammals, as you all know, present an exceptional challenge because they travel globally and are often only at the surface for a very brief period of time. The Argo satellite system remains the only viable way to telemeter data to a satellite. With Iridium and other Globalstar and those satellite systems, they are too large and too power hungry, even still, for the requirements of the marine mammal tracking community. Locations can be achieved either by the Argos Doppler processing, which has been in use since the 1980s, or by using FastLoc, which is a specialized acquisition and onboard processing of GPS signals that allows for the very rapid location of your animals. However, there is another emerging technology called RAFOS, which will receive acoustic systems from moored sound sources, allowing the triangulation of the geographic position of the animal while it is underwater. The range of these tiny receivers can be over 100 kilometers. So it's a bit like the acoustic arrays that is used for fish tracking, but uses the SOFAR sound system layer so that the acoustic signals can travel much farther. So this is just a quick diagram of how the triangulation would work, and in theory, it should be able to place an animal within tens of meters in 3D. Attached is the documentation for the initial studies, and Wildlife Computers will be working with the RAFOS team to try to bring the technology to the next level. So you know where your animal's, where it is. Now what is it doing? The development of tags depends upon the development and advances in the technology of consumer electronics. We need components that are both readily available and relatively inexpensive. Sensors and cameras are becoming smaller and more low-powered, making it feasible to incorporate them into tags. Large memory storage allows integrated, high-frequency sampling of many sensors. Robin Baird, whose talk will be following mine, will give examples of such complex tags. The animals respond to the environment, and as we all know, our environment is changing. Questions regarding the changes we're seeing in the ocean structure and how animals respond to these changes drove the need to develop sensors that could accurately and precisely measure oceanographic parameters. Robin's talk also will touch on some tags that look at these multiple parameters. But one of the things that Wildlife Computers has worked on in the last year is to develop a conductivity sensor which would yield salinity measurements. These measurements are an important parameter in understanding ocean structure. We've developed an electrode-based CTD tag that alleviates the near-field influences of the animal's body that has been plaguing some of the current CTD designs. These CTD tags have been successfully deployed for 2 seasons now on bowhead whales off of Greenland. The first season, 5 tags were deployed, the second season, 10. There are about 4 more tags to go out on narwhal later this year. And here in this photograph, you can see one of the CTD tags deployed on the bowhead whales. Along with the conductivity, temperature, and depth readings, all which are taken at high precision oceanographic quality, GPS locations are affixed to each of the casts. The whales are turning out to make excellent oceanographic sampling platforms traveling hundreds of kilometers where it is difficult to do surveys. So we've talked about where the animal is, what it's doing, and what's in the environment. Another question now is, who is interacting with it? Knowledge of the animal's individual behavior is great, but how do the different species interact? Who preys upon whom? So as an example, we have developed 2 specialized tags to help answer that question. First, I'm going to talk about stellar sea lions. Everybody has heard about the decline in the numbers. They've been rapidly disappearing in parts of Alaska over the years, yet only rarely did anyone ever see or sample dead sea lions. Live sea lions studied in the summer, when they haul out, would breed and seem to have healthy pups. Dr. Marcus Horning approached us about 10 years ago to collaborate to develop what was called a life history tag to determine where, when, and why the sea lions die. Here is a picture of the 2nd generation of life history tag. This one is small enough to go into stellar sea lion pups. We're currently working to improve this tag, further miniaturize this tag so they might be able to be deployed in fur seals. These LHX tags to date have given a surprising indication that the cause of sea lion mortality is often predation. As of a couple of years ago, 17 tags reported in. Of the 17, 15 gave enough data to determine the probable cause of mortality. And it was surprising that of all 15, the reason appeared to be a catastrophic event or predation. And besides the usual suspects of killer whales or other marine mammals, the data indicated a previously unsuspected predator, the Pacific sleeper shark. Another special tag development that addressed the question of the interactions of harbor seals and salmon smolt. This was just a pilot project, and working with the University of British Columbia, some researchers there, we developed a specialized RFID reader that was calibrated to pick up RFID tags that are implanted into smolt. And for those of you who may not be aware, an RFID tag is a passive tag that returns a signal, a coded signal, when it's pinged at by an active signal. So these RFID tags were implanted in smolt and then calibrated in captive studies so that the reader would pick up the ID of the tag when the smolt came close to a seal's head. The RFID reader was mounted on the harbor seal's head, and the seals were also outfitted with a buoyant backpack that had a 3D accelerometer and GPS recorder. 20 seals were tagged, and about 40,000 smolt, RFID tagged smolt, were released. The Argo system was used to track the seals as well and to help in the backpacks, recovery of the backpacks. The yellow dots indicate where an RFID tagged salmon was eaten. So it appears to have clustered around certain areas. The other dots indicated non-tagged, indicated the locations of the seals when they were not consuming the RFID tagged smolt. So besides kind of the internals of the tag, the externals are really important. Tag shapes have advanced a long way from the initial tubes and bricks that some of you may have used in the past. Though the most common way to attach a tag still is gluing it onto a seal's fur. The tags, however, have become smaller and more hydrodynamic so that they would impact the seal's behavior the least. Also, there are many various sizes and shapes for different animals. Cetacean tags provide an even greater challenge, and Robin will touch base on those. In some cases where animals have regular haul-out sites, researchers can recover a tag. A device called the payload recovery device allows a tag to be decoupled from the seal without disturbing the colony. These tags have been tested now for several years in the field. And finally, when you can't get the tag back, you have to telemeter it through the Argo satellite system. I'll say again that the Argo satellite system remains the primary way of getting data back from marine animals at sea because of its ability to receive data globally from small, low-powered tags. The Internet of Things, which is growing, is driving the development of new nanosatellite constellations such as Astrocast and Argos Next, but these are still years away from becoming operational. We can augment the reception of Argos data through the use of land-based motes, which also pick up the transmission and record them from Argos tags if it's within line of sight of the receiving stations. About 10 motes have been deployed worldwide. Some have been used for for studies around naval exercises. Others are in very remote places, such as this one that was deployed on Vancouver Island. It was needed because the channel was a east-west channel and there was very poor coverage north-south. And another mote was deployed on Ningaloo Island because they needed the greater data throughput. Finally, you've collected all this data, now you need to make the most of what you've got. researchers are no longer a single researcher working in a lab. There's collaborations that go on all the time and shared routines to analyze the data. Manufacturers all have our own data portals, and there are multiple global networks to archive data for collaboration. Some of them are the ATN network in the US, IMOS in Australia. In Europe, there is EO for wildlife, and this increases the value of the collected data, allowing for statistical analysis and modeling. Thank you.
We have time for a couple of questions. Francis.
For that. As you know, one of the concerns from a sort of vet's perspective on impacts of tags on animals is finding a way to have a tag completely internalized or not having a breach of the skin. And I know we've talked a lot in the past, but I was wondering if you'd had any sort of more breakthroughs or ideas that were hatching about how to do that, how to get transmission to a satellite through blubber.
We're actually working with that with Marcus Horning for the LHX tags and trying to up the power of the transmitter and find the right tuning such that we can get a signal through the animal when it is implanted. We've had some success implanting transmitters in pythons, for example, and we did work with Russ Andrews trying to put them in seals, and at that time the transmitter technology was still emerging, and it was kind of cute to see Russ walking around with blubber on his back trying to pretend he was a seal. But so we are working on that, and as transmitters become more powerful and we learn more about how to tune them, it's a potential.
Okay. Thank you very much. We will have time for questions at the end of the session as well. Excuse me. Hello. Keep going. Keep going left. There we go. All right. Our next speaker is our own Robin Baird, also from Cascadia Research Collective.
Thanks very much. And thanks, Melinda, for that presentation. It's really complimentary, I think. Now, it's probably needless to say that there's a whole variety of questions and management needs that are best answered by tag data. And these include things like looking at responses of individuals to vessel noise, examples with right whales, beaked whales, and blue whales. Call rates of individuals for interpreting data from acoustic surveys or from bottom-mounted hydrophone arrays or bottom-mounted single hydrophones. And this has been a topic for beaked whales and blue whales, among others. Responses of individuals to mid-frequency active sonar. There's been a number of studies on beaked whales, but also on other species. Overlap with fisheries and actual interactions with long-line gear that may be happening quite far offshore, and there's a couple studies that have been doing that both in the Pacific and in the Atlantic. And population structure range and high density areas in relation to critical habitat, and that's some of the work that I've been involved with in Hawaii, and there's other examples with right whales and a number of other species. Now, there's a wide variety of methods for deploying these types of tags on cetaceans. That includes some that are basically surgically attached to animals that are restrained in some way, and then other tags that are deployed remotely with either suction cups or darts. And I'm going to talk a little bit about these different deployment methods. Now, a little bit of history of the first radio tags that were deployed on cetaceans. From what I've been able to find, Bill Watkins and Bill Cheville actually tagged a North Atlantic right whale deploying a tag from a helicopter back in 1962 and again in 1965. Although they didn't actually get any signals from the tag, in part because they had a whole bunch of other tags in the helicopter that were transmitting at the time. Live captures for deployments, I think, started in 1968, and And I think Randy actually was involved in live captures of bottlenose dolphins in perhaps 71, 75, on either animals that were deliberately captured for tagging or animals that live stranded and were released and tagged upon release. Jeff Goodyear, I think, was the first person who used suction cup attached tags on cetaceans in the wild. Remote deployments on humpback whales in 1981, but those types of tags also basically allowed for tagging small cetaceans without capturing them for the first time. It was my first involvement with tagging was in 1991, deploying suction cup attached tags on killer whales. But probably the largest recent advance in tagging techniques for cetaceans came about through remotely deployed dart attached tags that were developed jointly by Russ Andrews with Wildlife Computers, the first deployment in 2004. And I brought along a couple of the darts that are currently being used for these if you're interested. They're short darts or long darts made of surgical grade titanium. Now, these tags are called limpet tags, low impact, minimally percutaneous external electronics tags. Even though the first deployment was in 2004 and we started using them in Hawaii and others started using them in 2006, that was just in the early sort of beta testing stage, and they didn't become widely commercially available until 2010. But these tags have allowed a much wider range of species and a much wider range of researchers and areas that they can be deployed in. This graph shows the cumulative number of species tagged with limpet tags over time, and they've now been deployed, as far as I can count, on 29 different species, 3 families of mysticetes and 4 families of odontocetes. Now, you'll notice in the first few years before that inflection point, the majority of the species that were tagged were relatively large species or relatively accessible species, and there's a few exceptions in terms of size. Some pygmy killer whales, melon-headed whales, and bottlenose dolphins were the relatively small species tagged in the early years. The more recent years and the slower increase has included a larger number of relatively small species, Stenella, Delphinus, Steno. Now these tags, the limitations on their application for smaller animals are really limited by the size of the target surface. They're deployed with a pneumatic projector or crossbow, and Melinda also brought one of the deployment arrows for the pneumatic projector here, which you can take a look at later, and also one of the limpet tags if you're interested. And for small animals, the target is just so small that it basically requires an expert marksman, to say the least, to be able to deploy those types of tags. And the future applicability of these tags is also limited by the approachability of species. They're deploying these tags typically at distances from between about 3 and 20 meters. And for successful attachment, you really have to have it very perpendicular to the fins. So a lot of species will probably never be approachable within those distances. Now, tags themselves stay on for varying periods. I've put 2 examples here. One of the smallest species we've tagged, pantropical spotted dolphins, median attachment duration of about 16 days, and false killer whales, median attachment of just over 40 days, but with a range of up to almost 200 days in false killer whales. And we've had one of these tags last for, I think, about 360 days on a pilot whale. So certainly not necessarily as long as we would all like, but long enough to get some quite useful information on habitat use and movements. Now, I'll show a little video here that shows deployment of one of these limpet tags on a false killer whale. And the other thing that the video illustrates is sort of the importance of combining the tag data with other data streams. In particular, in our case, photo identification information that we can use to look at the resighting history of the individual and potentially which population it's from. This particular camera setup that you can see here also has a green dot laser photogrammetry system, but various let me get that back in. various other types of information, for example, collecting biopsy samples for genetics or reproductive status, or using drones to look at body condition. Now, this false killer whale is obviously one of the more easily approached species. It's actually coming over and going underneath the bow. You can see the Daniel Webster in the bow with the pneumatic projector, and the animal's going to come up on the other side of the boat and have a tag deployment. So that's one of the better case scenarios, relatively calm seas, relatively cooperative animals. And so in most circumstances, tag deployments on this species, for example, go relatively smoothly in terms of the number of tags we're able to get out when we find a group. But there are lots of other species that, even though we've deployed tags on them, the opportunities to deploy them are few and far between. Now, another trend in tag developments and tag approaches for cetaceans is, as Melinda mentioned, just an increased commercial availability and an increased range of sensors. A number of years ago, when I first started using Wildlife Computers products, we would basically take one of their time depth recorders and build our own syntactic foam housing in my kitchen. And there were just no commercial vendors available that were selling complete packages effectively. Now there are quite a few out there. This particular system is including a dual camera system as well as a hydrophone. These are tags that are currently being used on baleen whales. John Kalambokidis from Cascadia and others are also still building their own tag bodies and putting together instruments from a wide variety of manufacturers to try to basically come up with their own custom instrument packages. On the top here is a tag that includes Wildlife Computers satellite transmitter, an ATS VHF transmitter, and a CATS accelerometer and magnetometer. Also has depth sensor, and it also has a fast lock GPS tag from Wildlife Computers. These are basically dart attached tags that are taking the idea of the digital acoustic tag, the D tag, which is an archival tag that is attached with suction cups, and trying to get longer duration information. So instead of just getting the 10 to 20 or 30 hours from a suction cup attached D tag, these are designed to record information, in this case the lower version, which has an acoustic tag for up to 3 weeks. And these types of tags are dart attached with a pole, given the size, and need to be recovered in order to get anything other than the Argos data back. But they're limited in their use, I think, in some ways, because when they come off the animal is entirely driven by random factors in terms of tissue retention around the darts. Wildlife Computers is working with Russ Andrews on an ONR-funded grant to come up with a consolidated dart-attached medium-term archival tag that has a remote release mechanism. And we actually deployed the first of these tags in a field project last month. They include a wide variety of sensors and have a transceiver for two-way communication, so you can communicate with the tag and basically send a release signal. I also included a drone photo here of one of these tags on a pilot whale, and the same individual taken from above water, because I think that not only do the drone images allow you to do things like assess body condition, but in this case it really puts the size of the tag in the context that you don't normally get from the surface photos. And you'll see on this pilot whale also has a very large remora on its peduncle. And these tags record acoustics up to 190 kilohertz sampling rate for up to 14 days. We put out 5 of these tags on shortfin pilot whales, used the remote release mechanism to get 1 of the tags back, and the 3 of the tags came off on their own, and just had one tag failure, communications failure for out of the 5. So given that it was a beta test, I think it was a pretty successful one. This slide shows an example of combination of information from this. If the speaker was working, I'd be able to play you some sound clips from it. but it includes the accelerometer data, the complete record of dive data, as well as a more detailed look at dive data, as well as the track of the animal from the FastLock GPS. So a couple other recent developments. There are ongoing efforts to improve tag design for suction cup tags, limpet tags, and the deep penetrating tags. I'm just showing one quick example from suction cup tags. This is work from Alex Shorter and his colleagues. Basically, there's a lot of efforts in the last few years to try to get smaller tags that have less impacts on the animals, and that's really occurring for all the different methods of tagging cetaceans at this stage. The last thing I want to mention is there's also been a increased effort, primarily funded by the NOP program and Office of Naval Research in the last few years, to follow up tagged animals to assess survival and reproduction. And this is an example from work that Alex Zerbini and Duke Robbins are doing with humpback whales in the Gulf of Maine. And I think these types of follow-up studies are critically important as these technologies are spread rapidly globally, and a lot more investigators are now using various types of invasive tags for cetaceans, and the follow-up information is a critical part for really understanding what the potential effects of those tags are. So I think that's it.
We have time for one question. Peter.
Just talk a little bit about the difference in working in different countries with tagging. Clearly, the United States has the MMPA and permitting processes. Do you think it's under control around the world, or are there any concerns about taking commercial tags and just using them willy-nilly or something like that?
Yeah, I definitely think that, well, I should say that there is an effort right now underway in order to come up with best practices, guidelines, and that there was a workshop last year, and that is something that's coming out. There's lots of concerns among the folks who initially started using the tags. When you think about it, particularly in terms of suction cup tags, are not obviously having any sort of major impact on an animal, but when you're remotely deploying a dart tag like this, if you don't have the skill or expertise, there's definitely a risk of injury to animals. So I think that there probably would be great benefit from having more widespread international guidelines developed and accepted.
Thank you very much, Robin. Our next speaker is Jeremy Sterling, who will be talking to us about sail drones. And why is it not coming up?
Just hit this right here.
I'll try that. That'll work. There you go.
All right, I'm filling in for Carrie Kuhn today. She couldn't make it. She's the lead on this project, sail drones in the Bering Sea. And we're using autonomous or unmanned vehicles to surveys to examine the relationships between northern fur seals and their prey. This project is a collaborative project. You can see a list of all the individuals that are involved, and also the different groups: PMEL, JASAL, Sail Drone. The Advanced Technology Group, Wildlife Computers, has been really helpful with this project. So, and it's really spearheaded by Calvin Morty, who has kind of helped bring this technology to the Alaska Fisheries Science Center. The first sail drone survey work that was done in the Bering Sea was in 2015. I'm going to be talking about 2016 and 2017. He came to us and asked us if we had any use for this type of technology. And of course, several of us in the Marine Mammal Group jumped at the opportunity. So for those who don't know yet what a sail drone is, it's an unmanned solar wind-powered sailing vehicle. It's powered by the wind, and the power source on board is all solar and batteries. And it also has a satellite link, and it transmits near real-time information from a variety of sensors to the people that are operating, the sail drone folks that are operating the vehicle. And here's a list of all the different sensors. There's quite a few, and if you want this, so you can actually see what it looks like up close, we can give it to you. But we're focused, the survey information for the Marine Mammal Lab really focused around the passive acoustics, listening primarily for right whales in the Bering Sea, but also using acoustics, sonar, or the SIMRAD system to measure the availability of resources in and around the areas where the fur seals were feeding. The motivation for this work, as many of you know, there's been a decline over a long period of time. Primarily to the right of that gray box is an unexplained portion of the St. Paul decline, which is shown by the black dots there. The gray box area is when they instituted the adult female harvest. And so a large portion of that decline can be explained by the harvest itself. And just for reference, for Boguslav, since we talked about it earlier, Tom talked about it, the red dots are the increase that's been going on there. But you can see the changes since the mid part of the 20th century, and then compared to 2005, these are shots taken from the same site. There's been a dramatic decline. The other part of the motivation is that there's some indication that St. Paul has been declining the most. Tom showed you an animation earlier which showed the summer conditions, the summer foraging. There were distinct regions that each of the islands were feeding at. But during the winter, all three of those eastern stock sites mixed throughout the North Pacific. So it's put emphasis on trying to understand what's going on within the habitat of the St. Paul Island animals. And so this Carrie's work is really focused on this summer portion. And here's that segregation that we saw in the animation. The survey area that we focused on is in the area in blue or purple. I'm not sure what it shows up there on the screen. That blue-purple area is the foraging area for the eastern or northern part of St. Paul Island, which is about half of the pup production occurs on that side. And then the southwest part of St. Paul is the red there, and then the green is St. George, which is an island 40 miles to the south. If you go further south to Boguslav, that's that turquoise color. And the focus of the work is on adult females, tracking them, surveying the availability of resources for them in and around where they're feeding, and how that translates into their pups' growth. In addition, our program has been really ticking off a lot of the conservation action narrative, the items listed in the conservation action narrative of the conservation plan. This particular study focuses on the action narratives that suggest that we should improve our knowledge of the numerical and functional relationships between fur seals, fisheries, and fish resources. And in addition, conduct oceanographic and fishery surveys based on fur seal habitat use, which hasn't been done. You know, get ship time is very expensive, as we all know. We haven't done this in a very long time. And with the sail drone, even though it's an unmanned vehicle, we got over 100 days of dedicated survey time, which was fantastic. So the sail drone mission in 2016 and 2017, their goal is to help understand how changes in the ocean are affecting weather and climate, and to monitor whales, fish, and fur seals in the Bering Sea. And because this is a new technology, really the first couple years were involved in engineering and testing the different types of oceanographic sensors and different sampling schemes, with the goal of collecting and transmitting near real-time data. Also to test and deploy a low-powered echo sounder. This whole system runs on batteries and solar, so they had to figure out how to put all these instruments together to get them to work and not interrupt each other. They tested a passive acoustic recorder to detect the presence of marine mammals, and really specifically to try to see if it would work with North Pacific right whales. And then relative to the fur seal work, map the vertical and horizontal distribution of walleye pollock. Walleye pollock is the primary prey for those animals on St. Paul feeding on the Bering Sea shelf. Some results from this. Basically, between 2016 and 2017, we had, in 2016, we had twice the effort. There were 2 sail drones, so 100 days of surveys, and 65 of those days covered the fur seal foraging core area. In 2017, we had 1 dedicated survey. There were 2 sail drones that went up to the Arctic. We had 36 days of effort in the fur seal foraging area, but the focus in 2017 was a little different in that we collaborated with Wildlife Computers, and the technology group bought some cameras with us, so we had focal follows. We had more effort or time focused on following individual seals that were recording video images of what they were eating and surveying the availability of resources in and around that area. So even though it wasn't as much as the 2016, it was a much finer scale look at what was going on. This just shows the survey area. We're focusing on that kind of middle shelf region. That's where the animals on the north and east side tend to go to. So in 2016, those are the 2 sail drones that surveyed. In 2017, those are the track line from the single sail drone. And this has been well known for a long time, but at the same time, we have fur seal behavior in and around these areas. This gives you an example of an echo sounder image that shows the vertical spatial variability in pollock. Old pollock like to eat young pollock, so they don't like to be close to each other. You can see that the age 0 pollock really aggregate near the mixed layer depth. If you had a CTD tag, you would see that was the mixed layer depth. And the adult pollock shown there is typically more on the bottom or in the mid-water. So that's the vertical distribution, and the spatial, the horizontal distribution varies, the age classes vary as well. In the left figure, you see that there's age 0 distribution, the high densities there in red. And then this is a survey in 2016. And then skip over to the next figure on the right. Those are where high densities of adult pollock were seen. And so for 2016, this is the spatial distribution of the animals that we tracked. There was 29 animals we tracked. And this shows that there was a correlation between the backscatter from the echo sounder and the number of hours spent. And this is all preliminary data, by the way. We're just getting into the nuts and bolts of this. And it coincided well with the age 0 distribution. There were 4, skip ahead here. If you look at the 2 figures on the right, the one on the left are what I just showed you, but on the right, there were 4 individuals that were feeding up to the northeast, and their diving was much deeper, and they were feeding they were focusing more on adult pollock. So this is how it looks when you put them both together. The top part shows diving. The diving data is there in the black line. The echo sounder data is in the background. This individual was really focused in on age 0 pollock, from pollock that was essentially spawned in the late winter and had grown to about 4 or 5 centimeters by that time. So we see that the diving shallower, the dives to the mixed layer increased, and Melinda's famous wiggle detection, the number of wiggles increased. And then when they're feeding on the larger pollock, they're obviously going down the bottom, and the number of dives decreased, the wiggles decreased. So like I mentioned before, in 2017, we did these focal follows. This figure shows the individuals that were tracked, which are the dots. And then the orange area is the highlighted sail drone survey area. And the sail drone was able to keep up with the animals, which was really exciting. So, and then the added level in 2017 is those focal follows. Those animals were also equipped with cameras. So to continue on with the theme of the small versus large pollock, I have some video to show you. So keep in mind we have all the behavior and accelerometry data. This is a collaboration with Wildlife Computers. to help identify prey capture events through accelerometry. So we have the availability of resources in that area as measured by the echo sounder, and then we also have the capture of these events from the individual animals themselves. And back home, we were collecting information on the pup growth. So let's see if I can do this. This might give some people vertigo. This is a fur seal feeding in a group of H0 pollock, a big mass of it. And just going around and picking them off kind of like us eating popcorn or something like that. And close to the surface, you can see it's kind of during the daytime. So that's the small pollock video, and then the real unlucky adult pollock. This animal's on the bottom right now, cruising along the bottom, and you'll see it'll stop. You'll see some brittle stars off, and then you'll see a very unlucky pollock that crosses into the screen. Gotcha. Okay. So we're collecting a lot of information from instruments that both Melinda talked about earlier, getting the accelerometry data to identify those capture events. All right. So in summary, sail drones have successfully mapped out the fur seal prey landscape. We've collected simultaneous tracking of fur seals to examine how they respond to food and the variability in prey resources. And our ultimate goal is to link these behavioral changes with measures of foraging and reproductive success. And our next steps involve the multi-divisional collaborations. We want to integrate this result with historical data, demographic data, and really get it into the larger ecosystem models of the Bering Sea. We're standardizing. We have 863 tracked individuals that go all the way back to the early 90s, and we've recorded over 1.5 million hours of fur seal tracking and somewhere in the order of greater than 4 million dives. Again, we're going to integrate this with the various surveys. There's a lot of good coverage in the Bering Sea, and also into the ecosystem models, specifically the FEAST model, which is a bioenergetic model of the Bering Sea, and the Seattle model, which is a multi-species stock assessment model. We're going to try to get at estimating the age-specific consumption of pollock so they can put that into their models. And that's it. Any questions?
Jeremy may need to leave before the question session, so if you have questions for him, let's try to get them to him now. Robert.
Yeah, Jeremy, cool stuff. Could you talk about how the sail drones navigate? I mean, it seems like there's a couple of different ways that they happen, but if you could talk about that and how do they follow the seals?
Yeah, so we would we'd work with Melinda in the Wildlife Computers portal and pull that information in and send it to the sail drone folks, and they would navigate it. But they are developing techniques like acoustically, where if you had some sort of pinger that the sail drone could pick up on the individual animal and follow it, they're talking about doing that. I don't know if they've done that yet or no. They're moving in that direction. Yeah. So it was very manual. Pull the data down the portal. Here's the locations. This is where the animal's going, and the sail drone would catch up to it pretty quick.
Can you program them to do tracks or transects kind of thing?
Oh yeah, so that's what. Yeah, it's daily. You can talk to it daily and tell it where to go, but you can also just put in, here's your survey grid. You can modify it at any time. It's pretty sophisticated. A lot of it's done from on the phone. So yeah, the sail drone guys are very helpful in trying to get this thing to work. It's been really nice.
Sam.
Fascinating talk, Jeremy. Very cool stuff. Can you talk a little bit about other diet studies that are being done on northern fur seal? So you have the specific information about what these particular seals are eating, and you can translate that into bioenergetics or measure pups back on the rookery. But is there that missing link of diet studies through scat or fatty acids or stable isotopes?
Well, what I didn't talk to is the kind of wide range of research that our program does do. We have a very active diet program that is scat-based, spew, because there's differences in scats and spews. Both samples are biased in different ways. Fatty acids and isotopes. There's been a lot of advancement in the isotope area that our program is actively doing. I don't know if there's anything I've left out, Rolf. Got it, okay.
More questions? All right. Thank you very much, Jeremy. It's fascinating.
Yeah, thanks.
Okay. Next, we're going to hear about the latest with passive acoustic monitoring with Catherine Burchak.
So thank you. I've been asked today to talk about passive acoustic monitoring, how we use it, what we've learned from it recently, and what we hope to do with it in the future. And I'm from the Alaska Fisheries Science Center, so my talk is going to be geared toward the Alaska region. So what is passive acoustic monitoring? Basically, it's studying marine mammals by listening to the sounds they make. And we do that typically with a hydrophone, which is a sensor that detects pressure variations caused by sound waves in the water. How do I get this to stop advancing?
Let's see.
It's like doing it automatically.
We don't want it to do that.
No.
All right. Try just doing it manually like that, see what happens.
Okay. All right. So these hydrophones can be fixed to a variety of different platforms. Some like dipping hydrophones have been used for years and years and years, cabled hydrophones or observatories, and also, wow, that is really just speeding right through. I'm going to end it for a moment.
So did you start it from here or from?
He just
I
did.
started it from down there.
Okay, that's just.
I don't have any sounds to play this time, so that part should go all right.
Try this, but
Okay.
I don't know if that.
That is what
Okay, so dipping hydrophones, cabled observatories, and the workhorse of the Alaska region, moorings, which are usually anchored, well, they are always anchored to the seafloor, and the top floats rest pretty far away from the sea surface to eliminate surface motion and noise from the surface. So those are all on fixed platforms. We're also able to attach hydrophones to a variety of mobile platforms, such as towed arrays from ships, sonobuoys that can be deployed from aircraft and also vessels, tags, which we've heard a lot about, thank you, and also autonomous vehicles, such as, it's still moving. Let's go back. Jeez, stay. So like the Slocum gliders shown here, and also the sail drone, which Jeremy gave a nice introduction to. So the benefits of passive acoustics is you can monitor multiple species at once, including pinnipeds, both toothed and baleen whales, and also other marine organisms such as fish and invertebrates. And also sound sources caused by humans, such as vessels and seismic air guns, and environmental noise like ice, rain, and earthquakes. The benefits are that you can record data all of the time, no matter what the sea state, visibility, or light conditions. So what have we learned so far? One of the greatest contributions of passive acoustics, I think, is that it can provide these very long-term time series of seasonal distribution of species. So here, this shows a 5-year record of bowhead whales from Barrow in the Beaufort Sea on this top row here, all the way down into the Bering Sea, which is the bottom row here. Each of these black lines is a day, and the height of that line is the percent of 10-minute time segments in that day that have the presence of that species, in this case, bowhead whale calling present. The blue lines here show the percent ice concentration from satellites in a 20-mile radius around each mooring. And one thing to note just quickly from this plot is that there's differences in the bowhead use around ice in the Beaufort region, where they're just passing through on their migrations in the fall and the spring, and in the Bering Sea, where they're actually wintering, where they're spending most of their time in the ice concentrations. Also, in this area here, just south of the Bering Strait, you can see there's huge interannual differences in their presence just in that one spot. Go back. Okay. Which is very important with the rapid changes of the climate up in the Arctic. This is another plot of, okay. This is another plot of the bowhead distribution. The other one showed it sequentially. This combines it all together and takes a median. So shown in green here is basically all those days that have darker green are each of those 5 years had bowheads detected on that day. And what this can be used for is to gain a better understanding of the seasonal ranges of these animals. So on the right is a plot of the distribution that's currently known of bowheads in the wintertime in the Bering Sea. And this is from satellite telemetry data from SIDA's group. And it shows the core use areas up here in the northern Bering Sea. And if you get into the 95th percentile range, it's still in the northern Bering Sea with just a little bit down in this area. And these green stars, these 5 green stars here, are out of that range for the wintertime. But what those 5 green stars are these mooring locations from M5 down to M2. And as you can see, there is bowheads presence pretty much every year from December through April, May in those areas. Another species for this, where this is really important, is the very endangered North Pacific right whale. I'm not going to be presenting that information because Jessica Krantz and Dana Wright will be presenting that on Thursday at this meeting. Sonobuoys also give us a better idea of seasonal ranges. For example, fin whales usually are not visually sighted north of the Point Hope area here. But sonobuoy detections have shown that at least some individuals make their way almost all the way up to Barrow. Another good thing for sonobuoys is when they're used on cruises where the biologists really need to find the whales to put tags on or take pictures. And in that case, they're really critical. Some sonobuoys can not only just record the sound from the species, but also the direction that sound's coming from. So when you deploy multiple sonobuoys in the water, you can cross-fix and get a location of the whale and direct the ship to it. And this was the case last year during the IWC cruise, where there was 9 sightings of North Pacific right whales in the Bering Sea, and over half of those were found through Jessica Krantz's efforts with sonobuoys. And in that case, they found 12 individuals. They were able to ID 12 individuals, which is nearly a third of the estimated population. This is another plot showing how this Pacificus can increase our understanding of seasonal ranges. This again is 5 winter seasons. You can see the 100% ice cover and a 5-year period from 2010 to 2015 again. And this is offshore in the Chukchi Sea, about 100 miles offshore. And here we have detections of beluga whales, walrus, and bearded seals. Now, all 3 of these marine mammals are very on the smaller side, and they can break through ice, but not very much, about 10 to 20 centimeters at a time. So the question is, how are they even there in the middle of the winter? So Sigurd Salo pulled up some ice images from satellites, and you can see that the ice is very degraded with lots of leads and polynyas in March. But the problem with satellite images is a lot of them are basically obscured by cloud cover. So it made me think that perhaps instead of you going and getting ice data to confirm that there's a reason that the species are there, that in contrast, you can use the presence of these species to actually act as a proxy for ice conditions when there aren't sensors out there getting ice thickness or satellites that can see what the ice conditions are. And getting into some more interesting results. This is from Susanna Blackwell's group, and Aaron can correct me because he was part of this group. They used these arrays of directional moored recorders in the Beaufort Sea, and they were able to localize almost 1 million bowhead calls, and they used a subset of those, about 100,000, that were basically in the same areas where they could get receive levels from airgun pulses. And what they found that as soon as the air guns were detectable, bowhead calling started to increase. And it increased until it hit a threshold level of 94 dB. And then that remained basically at a plateau until it reached another threshold of about 30 dB higher, in which case basically all calling ceased. And they found this from air guns. And then in 2017, they wrote another paper that basically found the same sort of double threshold effect from the tonal noises from drilling rigs. And Susanna's group also, from these localizations, were able to show that bowhead whales have directional calling. And why this is interesting is that typically people assume that whales make really deep, low sounds, and those sounds are so large that they carry kind of uniformly away from the animal. And what this has shown is that as the whales are moving away, there's actually more sound coming from the front of them than the back of them, contrary to what everybody has assumed in the past. Manolo Castellotti's group has been able to combine two different sensors, one that's just listening for sounds and another that's listening for echolocation clips, in conjunction with looking at behavior visually, and have hypothesized that it's possible to tell when beluga whales are feeding, in which case basically there's very few social calls. There's a lot of echolocation occurring in trains with this terminal buzz that's occurring. And what's interesting about this is that you can go now back into those long-term recordings and not only say that there were belugas there, but be able to say that they were feeding. And this ties in with the fact that most of these platforms that hydrophones are deployed on now have some sort of oceanographic data collection. And this is from Kate Stafford, who showed that up in the Barrow Canyon area, excuse me, that it seems like the strong Alaska coastal currents are bringing more beluga into the area, both more detections from her recorder and also from aerial surveys. And she hypothesized that this current is bringing more feeding opportunities. So passive acoustics can also be used to differentiate populations. In the Arctic, it's been used for minke whales and also fin whales from Julian Delarue's group from JASCO. And also Aaron Thode's got a very fancy algorithm that can use a single hydrophone to determine the depth of a calling animal. And his work has shown that bowhead whales seemed that they have an upcall that's just the same as the North Pacific right whale upcall, and it seems to be produced at a deeper depth than the right whales. And why this is important is that in the Bering Sea, both these species seem to have overlapping areas that it's really hard to tell apart because these calls are so similar. So it's another way to actually be able to say for sure it's one species or the other. And although this isn't really passive acoustic like I've mentioned in the introduction, it's a really cool project with Manolo Castellotti being able to measure in the field wild beluga hearing as part of a study that was capturing beluga whales to do health assessments. And what they use is the auditory evoked potentials, which is basically they play a sound to the whale and then they're measuring their brain waves to see if the whale's registered, whether it's heard it or not. And this is super non-invasive. Basically, that's what human babies are, they get, when they're newborn, they get hearing tests with the same method. And what he was able to show with the 7 whales that they collected this data from is that their hearing curve fit pretty much what they were getting from captive individuals. So future directions, we hope to just continue to collect these long-term data. And I can't emphasize more that archive data don't expire. They can always be reprocessed. So as we get more information on population differentiation and behavior context, or even know this fish species makes a certain call, we can go back through all these data and be able to get more results and more answers. And also to be able to study noise effects. Excuse me. This is a study we're doing with Audubon Alaska on vessel traffic. And this is just another long-term data series that's showing North Pacific right whale presence in Unimak Pass, which is a very high-traffic vessel area. And you can see there's not that many North Pacific right whales detected, but these light gray bars in the back are days, and that's the number of vessels detected on those the the number of 10-minute bins in that day with vessels detected. You can see from August of 2009 all the way through August of 2015, the vessels really never stopped. So it's really important to know how these animals are reacting to the noise and the human presence. One thing that passive acoustics cannot do right now is abundance estimates, because we can't count non-vocalizing animals, and we don't know how many are calling. But for visual observers, they don't get to see the whales out of the water all the time. They have developed statistical methods to be able to go in and know how many they're missing. And that is the new field that has basically been developing over the past several years. And this is a paper by Tiago Marques and company, which was just basically a proof of concept to get people thinking about this and be able to get more people to gather data to be able to make this happen. And as we get more data to fill in these dots, we can go back through all those archived data and be able to get abundance estimates from way in the past. And finally, as new technology advances, we'll be able to do more cooler, more interesting things. One thing is to be able to tow a hydrophone array behind the sail drone to reduce noise. Other is to maybe just make a fleet of these autonomous vehicles go out and localize on animals. But just to conclude, one thing to keep in mind is no matter how many whiz-bang things we've got, sometimes just dipping a hydrophone into the water, listening and watching will provide a lot of information. So, thank you.
Thank you very much. We'll have time for questions after the last talk here. Scott Baker will talk to us next about environmental DNA.
Thank you, Randy. It's here, it's there, it's in your hair. Environmental DNA, it's everywhere. And if so, can we harness it to help us improve our ability to detect and to identify species of cetaceans? Short answer is yes. Long answer is but. So species of identification of cetaceans is uncertain, certainly at sea and certainly to many of us. There are now 89 plus accepted described species of cetaceans and probably more on the way. Beaked whales are a particularly challenging group. There are now 23 known species, 4 new species validated just in the last decade. They are rare, elusive, and cryptic, or cryptic. Some never seen alive, unknown population structure, limited morphological characters, only the shape and position of teeth in mature males for most species. And they are susceptible to anthropogenic sound, particularly Navy sonar. Confident identification of cetaceans requires DNA barcoding, methods that we adopted for our studies of market samples more than 25 years ago, focusing on the control region of the mitochondrial DNA as a primary identifier of species, but also providing information on stock differentiation. So a single sequence of mitochondrial DNA from the control region compared to a reference database allows us confident identification of that unknown sample. This is possible because of the accumulation of reference data sets representing all 89 of those accepted species, and a shout out to the Marine Mammal Commission who helped support the development of the program DNA surveillance and development of that reference database, which includes about 400 sequences of the control region and about 264 from the cytochrome B. So almost every species of cetacean is represented by more than one sequence, capturing some of that variation within species. Collecting genetic samples of cetaceans, though, remains challenging. Of course, we can wait for them to die and throw themselves up onto the beach, or we can go out and kill them, or we can collect a biopsy sample, or we can collect fecal material. But in most of these cases, with the exception of the biopsy sample and scientific whaling, it's opportunistic. It comes at the discretion of the whale when we need to be able to sample systematically. So what about environmental DNA, or eDNA as it's now known? Well, this is now a well-accepted methodology, particularly in the freshwater aquatic system, with references going back now probably more than 15 years, and I'd say a general acceptance of the value, particularly for the study of rare, cryptic, or invasive species in freshwater, now maybe more than a decade. But the first report of the use of this for identification of marine megafauna, or at least of marine mammals, was in 2012. And I credit Andy Foote with inspiring several of us to write proposals to try to continue or expand this work. So the objective of our study that's been funded over the last 2 years by the Office of Naval Research has been to characterize and optimize eDNA detection of cetaceans. There have been 2 phases of this. Phase 1, where we focused on inshore sampling using a new methodology, droplet digital PCR, and conventional DNA barcoding. And then the phase 2, open ocean sampling with, again, the droplet digital PCR, but using now a meta barcoding methodology and next generation sequencing with Illumina MiSeq. So here in phase 1, the objective was to detect the eDNA plume in inland waters. And we can imagine that this eDNA plume has a number of sources. It comes from the spout, epithelial cells shed from the lungs, maybe white blood cells. It's coming from skin sloughed off the whales as they move through the water. And of course, it's coming from excrement. Killer whales of the Salish Sea or the Puget Sound provided a practical natural experiment for assessing the limits of this eDNA plume because of the predictable seasonal occurrence, the relatively calm inshore waters, and the diagnostic genetic differences of known ecotypes, particularly the southern resident ecotype, an ecotype that we've never had in our laboratory, so it was a useful control for any potential contamination. So our methodology kind of goes in a clockwise direction, starting with the collection of the seawater. We moved into position more than 200 meters behind the passage of whales, begin collecting at time 0, and then every 15 minutes for up to 2 hours afterwards. We collected 2 liters of water, 1 from each side of the boat, but it was just for a replication. We filtered the water through 0.4 micron polycarbonate filters. We stored those filters while we were in the field. We then extracted them back in the lab. One of our surprises, or unpleasant surprise, was the degree of inhibitors. That is, these are organic compounds that inhibit conventional PCR. We then adopted this powerful new technology, droplet digital PCR, to quantify that DNA. Then we were able to re-amplify and sequence the DNA using conventional methods. So droplet digital PCR, this is a method that takes a single PCR reaction, typically 20 microliters, fractionates it using an oil emulsion into more than 20,000 nanodrops, each of which becomes a PCR reaction. That PCR reaction, or the presence of your probe, your fluorescent probe, is then read through a flow cytometer for each one of those drops, and it tells you whether there was a positive or a negative. The intent here is to look at low copy numbers of DNA, so the presence or absence of that fluorescence in those droplets can be modeled with a Poisson distribution and tell you something about the absolute copy number, that is, the absolute number of DNA molecules you have in your sample. Okay, so that was a lot, but here's an example to give you a sense of the visualization of these data. So this was a single encounter with a group of killer whales off of Vancouver Island. We collected 5 serial samples, so again, starting at time 0, every 15 minutes, so last sample after 1 hour after the passage of the whales. We detected eDNA in each of these 5 serial samples, although with some considerable variation. So you see on your right the Quantisoft image or analysis of the droplet digital PCR. And you can see each sample was run in duplicate, and the estimated copy number is shown there at the bottom. So at the first sample, there were 4.5 or 2.5 copies per microliter that were estimated, and that's based on those fluorescent droplets, the ones that are shown in blue. The black droplets are the ones that were negative. So there's nearly 20,000 negatives, and there might be 40 or 50 positive drops. The next sample, collected 15 minutes later, you can see had a much lower number of droplets, a number of positives. Then it went up again, down again, down again, down again. But in total, there were all 5 of these serial samples had detectable copy numbers. and 3 of these were confirmed by reamplification and conventional DNA sequencing. So the conclusion from phase 1, which has now been published in a recent volume of the Frontiers in Marine Science, is that the power of droplet, there is considerable power to the droplet digital PCR for quantifying environmental DNA. We can confirm the reamplification for eDNA barcoding, giving us both species ID and ecotype ID. So we were able to identify the southern resident killer whales in our sample, as we expected based on reports from whale watchers and from acoustic signals. Confirms that the detection of eDNA plume can persist for up to 2 hours with 4 to 5 kilometer movement of water column. And I'm sorry, I failed to show you the positions of the boat, which were documented by a GPS drogue showing you that we moved almost 4 kilometers during that hour due to tidal currents in the Straits of Juan de Fuca. We've now moved on and completed the primary analysis of the open ocean sampling with visual and acoustic detection. And this has been done through a series of collaborations with different groups, 2 of which were working in Hawaii, including Robin Baird and his group doing what we refer to as surface sampling for social odontocetes, as well as deep water sampling in Hawaii during the High Seas program coordinated by Aaron Olson. We had 2 collaborations with collections from the Southern California Naval Range, one from the shelf of the eastern seaboard with Danny Cholowak and Sal Sergio, and one from the Bering Sea with Olga Filatova at Moscow State University. And I'll take just a minute to give you a sense of the results from the Bering Sea collaboration. So for the meta barcoding from these open ocean communities of cetaceans, we adopted this next generation sequencing. So the methodologies are pretty much similar up to the point of the droplet digital PCR for the purposes of quantifying the environmental DNA, but then we would take the samples that were positive and we would break the oil emulsion, re-amplify, and sequence these with next generation sequencing methodology. This gives us hundreds of thousands or even hundreds of millions of different reads, each of which represents one of those variants or haplotypes of cetaceans. We then analyze this using a program that basically stitches these together and tells us how many variants we have, and then identify each of those variants by a search of GenBank, the National Repository for Sequences, or using DNA surveillance, our taxon-specific reference database. And the intent here is to be able to identify multiple variants as opposed to the initial study of the killer whales where we used taxon-specific primers to target killer whales. So the question here was the potential to identify the 2 different forms of Baird's beaked whales, or the new species of beaked whale, the black form, or karasu. There are only a small number of these specimens of this karasu, one of which was collected in the eastern Aleutian Islands, and others of which collected from the north coast of Hokkaido. So there's a vast area of the North Pacific, particularly the Bering Sea, where we presume this black form may exist. Olga's work is in the Commander Islands, which seem like a likely area for this karasu, or even an overlap of the karasu and the gray form. And you can see a little bit about the reference information on the haplotypes of the gray form and the black form in that diagram below. So we know not only about the genetic differences between these, but also something about the variation within species. So the results, the MySequence barcoding gave us 77,000 high-quality merged reads. This confirmed that these were all gray form and that they matched 2 of the published haplotypes or variants. So we see 52,000 of these copies were a G1 haplotype, 20,000 of the copies were a G5. Others included variants that may or may not be sequencing artifacts, or they may represent previously undescribed variation within the gray form. So in conclusion, environmental DNA represents a powerful tool for the detection and identification of cetaceans, basically adding to the hierarchy of methods for genetic sampling. To put the sensitivity of this methodology on an organismal scale, we calculated that we can detect 1 128 millionth of a small biopsy sample from the 2 liters of seawater. So you can see perhaps from the arrow the location of a biopsy sample of this Maui dolphin. It's a very small biopsy sample, and this is approximately 128 millionth of that is detectable. But the quality and quantity of E8 DNA are not sufficient to replace biopsy sampling. So in my view, this is a powerful methodology, particularly for species that are difficult to biopsy sample, but it does not replace the genomic quality or archival quality of a biopsy sample. But for some species that are not necessarily elusive, but more evasive, it may well be a very powerful methodology for collecting information at the population level, and I think you'll hear more about this tomorrow from Kim Parsons. And with that, I'll thank Mike Weiss and ONR and our many collaborators. Thank you.
Thank you. We've got time for questions for Scott, then we'll open it up to the rest of the group.
Scott, it seems like eDNA has a lot of potential here in the near future. I'm curious about one of your last statements that eDNA is not going to replace biopsy sampling. Could you expand a little bit more on that? I mean, at this point, I'm assuming that eDNA wouldn't be sufficient for doing genetic marker capture. Right. If you could just talk a little bit more about
with current methodology, we're really replicating a DNA barcode. So it's species-level information, and then using the control region, which has quite a bit of intraspecific variation, perhaps stock information or ecotype information. But we're not going to get at this time individual-level genotyping. Now, again, there's such rapid development in technology that I would never say never. But right now, I think it's also I think that's going to be its primary power.
Peter?
Yes, Peter.
Yeah, so what is its primary power right now? Its presence or absence of species? I mean,
so I think
where is it now and where is it going?
Certainly there's power in opportunistic sampling and confirming your species ID. So moving into the proximity of an individual or group, particularly one of uncertain taxonomy or uncertain identification, and collecting seawater in the hopes that you can gather the eDNA for identification. The information that we've collected on that serial sampling is also beginning to give us information that may allow us to do a spatial sampling as well. I think that's still a bit ambitious, but it's certainly a direction we might be able to go so that we'd have a complementary tool for acoustic detection or visual detection, either from planes or vessels, to be able to overlay an environmental DNA identification or detection grid. So both spatial, temporal, and opportunistic. Yes, Randy.
Can you just quickly tell us the take-home message about your Baird's beaked whale study? You found only the gray form.
Only the gray form, which wasn't a surprise given that Olga's quite familiar with the gray form and I think would have recognized the black form. But that being said, I think there's a chance to go back and to perhaps look more specifically for the black form. Maybe again with acoustic detection to put us in the proximity, although I'm not sure that the black form has been acoustically characterized.
What's the sort of relative cost for doing these kind of analyses?
So that's a bit of the but, yeah. The straightforward, conventional PCR and sequencing is probably on the order of a few dollars a sample. You move into the droplet digital PCR, which is fairly expensive because of industrial costs, and you go into next generation sequencing, and you're looking at probably a few $100s per experiment. Now, in our case, we multiplex the sample, so we were able to run a number of experiments on a single MySeq channel, and so that reduced costs. So there's potential to get this down, to get it down to just a few dollars a sample.
Francis?
Kind of a little wild and hairy. But obviously, here you're getting little bits of DNA out of vast amounts of water. Has there been much thought about trying to get bits of DNA off fishing gear or ship hulls or things like that?
Oh, sure, yeah. I mean, there's certainly a lot of sources of tissue for DNA. And I think there was some discussion about putting cheese graters on the bow of cruise ships in Alaska so that when they hit a whale, which they will do, there's a trace left behind to be able to identify the whale. But this is working with things that are not visible. There's no visible particle in any of the samples that we processed. So this is another order of magnitude of a dilution effect.
Aaron?
How long does DNA survive in seawater? I mean, I think you have a dispersion effect here, but there's also probably a lifetime effect.
Yeah, there's a degradation issue. We don't know. I mean, there have been some laboratory tests of this, and they're looking at things like on the order of a day before probably fully degraded, but it's probably going to depend a lot on temperature, sunlight, and microbial community as well, because that's what's eating up the DNA. So we were a little surprised that we could detect it for 2 hours in Puget Sound, and that was the limit of our patience. Now, looking back on it, we should have done even longer serial samples to get a better sense. But there's probably a dilution. Well, there's certainly a dilution effect going on, and then there's also the degradation effect going on. And there's going to be a lot of variation in that.
Samantha?
This is kind of a follow-up from Francis's question. Are there any organisms that might help you? Like in terrestrial systems, they use leeches, for example. Is there anything in the marine system that could be an equivalent?
That's an interesting idea. Morays, cookie cutter sharks. Sharks. I mean, we have actually been able to get tissue out of the stomach of a shark to identify a Maui or Hector's dolphin years ago. But I think that's a possibility. You have to get a good animal trainer for perhaps a guillemot or a shag. Patty?
I'm wondering what you think it's going to take to go from presence-absence detection to actually, population frequency type data for stock structure, for example.
I think you'll hear a little more about that from Kim. For our purposes, the Baird's beaked whale was fairly instructive. That is, we got some sense of a potential quantification of the relationship of those two different haplotypes. That being said, we had pooled a number of samples which were perhaps the probably different social groups of Baird's beaked whale. But I think there's certainly potential for it. What the standard error or confidence limits might be, I'm not sure, and I suspect that's going to be highly variable as well.
We have time for one more question in this session.
Yes, Jason.
Yeah, I'm just following on some of Aaron's question, I guess, about persistence and dispersion of the DNA. I guess I'm curious to know what you're thinking about the use for the quantification. It's a kind of an unusual type of quantity that you're measuring, and how that might inform inferences about when animals were places and densities and that kind of thing.
Yeah, I think it's going to be very case dependent. So our interest was in relating that quantification to our success with the sequencing. At least that was our initial, was there a lower threshold below which we could detect, but we perhaps not sequence reliably. And we did get some information on that. How we might model this if we moved into, again, a spatial kind of context, I think we still got a bit of work to do. That being said, we do have a proposal into ONR to try to do this, at least a first cut in Cape Cod Bay for right whales. We think because of the extensive information on location, because of a system of sample collection that they already have, that the Center for Coastal Study already has in effect, that we might be able to piggyback on that.
Okay, thanks. Yep. I'd like to thank all of our speakers for this afternoon. We've got some amazing new toys in the toolbox, and it was really great to hear about it. I think at this point, we turn it over to Daryl.
Okay, thank you. So we're at the end of the day, and we have set up the potential for anyone from the public to comment if they haven't, or to make some formal statement. So let me see if there is anything anybody in the audience here wants to comment on, or ask questions of us or any of the people on the panels. Yeah, I guess the next question is, since we have had people online, is there anyone online right now that might have any? Vicky, are you there?
This question is from Gay Sheffield, and she said, tag share is a concern to the subsistence communities in Alaska regarding tagging programs. How is this issue being addressed in pinnipeds and cetacean tagging programs that operate in Arctic waters?
Could you repeat it, and do you know who it was targeted to?
It came up around the time of Melinda Holland and Robin Baird, and I'll repeat it again. So tag share is a concern for the subsistence communities in Alaska regarding tagging programs. How is this issue being addressed in pinnipeds and cetacean tagging programs that operate in Arctic waters?
Thank you.
Well, I
Alex?
don't work in Arctic waters. I can speak a little bit about the tag shear problem.
Alex, would you just say your name and
my
your
name is Alex
affiliation?
Urbini. I work with the Alaska Fisheries Science Center, with Cascadia Research, and also with Marie Cotel. So what I was going to mention is there was a study that was funded by ONR where Michael Moore and I were involved. There's a paper published. I can bring that up. I don't remember. I don't have it right here in front of me. What we did was we tried to measure shear on a carcass of small dolphins and try to extrapolate that to large whales. And you can go back and look at the paper. What we determined is that shear changes according to the position of the body. So if lower on the body, shear is of greater magnitude if you than higher up on the midline of the body. So I'm involved in implantable tagging. I'm involved in using tags that penetrate the blubber muscle interface. And our way to minimize trauma is to try to deploy the tags close to the midline of the body. That's where shearing is the least pronounced. In that paper, you'll see that the extent, if you extrapolate shearing that we measured on the carcass and has its limitation to a large whale, if a tag is deployed close to the midline of the body, the shear, the range of the muscle blubber movement is equivalent to the diameter of the tag. So it's not very pronounced compared to other parts of the body. So one way to minimize it is to make sure that you target the right areas on the body of the animals. But again, I'm not involved in Arctic research. I don't know how that's done in Arctic research. But this is a way to, at least partially address that problem.
But why are they worried about shear? Robert, do you know? You're reducing the fitness of the whales, or what?
So I'm not sure exactly why Gay asked about shear, but there is a concern among the native community in Alaska, but other places in the Arctic as well, about the impacts of tags generally on marine mammals. There's both this physical component to things, is it causing an impact to whale survival or reproduction? But there's also an ethical concern as well that that many Native people feel like tagging whales or tagging seals is like playing with your food, and it's being disrespectful to the animals. So there's been a vast amount of hesitance to capture and tag animals for those reasons. But people have been willing to go along with it because the information that comes back from those animals provides new insights to all of us, to scientists and hunters, about what the animals are doing.
So in part, it's a communication issue, making sure that those who undertake research using these kinds of technologies gets back to the communities and discusses with them, first off, that they're going to use them, why, what the results might show, and then following up afterwards with the results and so on. Yeah?
Yeah, thanks for all that, Mike. That I think in part it's a communication issue for all of those components that you said. I would maybe rephrase one of the first or second points in that instead of saying we're going to use this to approach the community, and says we have some questions that we think they match with what you're doing. Here's one way to do that. Is this an okay approach? I think there needs to be a willingness to modify approaches. If tagging is a major concern to elders, to hunters, try to figure out if there's other ways to get that information or figure out what the best ways are to approach the question. Some of the work that Alex is talking about, I think, goes a long way to help trying to reduce some of the physical impacts to animals. And I think that is certainly helpful in Arctic communities and with hunters.
Yeah, I was just going to comment on that. I personally have been involved in a research to improve these types of tags, the tags that penetrate the blubber or the blubber muscle interfacing going to the muscle. We don't have the results published yet. I've presented this before, and in my opinion, and I think other people would agree with me, the tags today are better than the tags were yesterday or a while ago, primarily because one of the issues these tags had were design flaws that resulted in breakage after tags were deployed on the animals. And currently, with the new tags, this problem has been minimized to a large extent and potentially even solved. I mean, I wish there was an opportunity to discuss this further with this group. I don't think we don't have the time here. But the tags are better today than they were a while ago, and the health and the risk, the health issues and the risk has been minimized. And obviously, there's a lot more development that can be done and should be done, but the new advances that have been made should be taken into account when considering, future studies and future tagging, not only in the Arctic but other parts of the world.
I think Robert is right on, and some of the concerns from the communities that do harvest these animals for food. One of the issues that we heard with walrus tagging was that some of the darts or implements would wash up on the beaches, and that was a concern to the hunters who find these debris basically on the waters and not knowing exactly what they were for was some of the concerns that I've heard. Especially during our walrus tagging for that, what's that one with Patrick? Patrick will be here tomorrow, Patrick Lemons, who may add more to this. But
okay, I guess if that's all we have on that end, then maybe what we'll do with time remaining is I'll ask the commissioners and committee members if you have any thoughts or follow-up from various things you've heard today that might lead to ideas, actions that we might take, if there's anything along those lines that you want to raise at this point. Go ahead, Mike.
Thank you, Chair. You and I discussed at a break that the two of us have lost track of the progress, shall we say, of the Marine Mammal Health Map Initiative, and shame on us. I think that the Commission and the staff need to have a discussion about where this is going and what might be done to keep that going. That is, after all, one of our strategic initiatives, one of our most important strategic initiatives, and if it's in difficulty, what can we do to help? And are there other agencies out there, perhaps in NOAA, that might be engaged to help us move it to the level we believe it deserves? So it's probably more of a recommendation to ourselves to follow through on this than to cite it as a major public concern, but it's certainly one of importance to you and me.
Yeah, I guess one of the things that crossed my mind is there anything we can do when we go to the Hill to offer up thoughts on if funding is an issue, might there be some way of seeking funding? And then I heard Sue say some ideas on how to best integrate this with observing programs and so forth. So I guess the question is are there actual things we could do that might keep this moving along? So I don't know that we'll come up with answers here, but it's something that I think we should keep talking about. Kind of along those same lines, one question that crossed my mind is, Francis, you had mentioned the funds in the UME program, and I forget exactly when money was last put in there. But is there some sort of formal process for seeking additional money, and what kind of time frame might you expect that money to become limiting to deal with UMEs?
Well, the money itself is within, I mean, it's an appropriation, so it's as everything else in Congress. As far as timeline, it's limiting now. I mean, the reality of towing a single whale for necropsy is, $5,000, and you necropsy each of the large North Atlantic right whales that died, and that's the end of it. And I think there's also the other point I didn't make was that NOAA has really taken the brunt of making sure there is money in that fund and also managing it, but in fact it's also being spent on Fish and Wildlife Service species. And there's never been money from Interior put into the fund. I'm looking at Jason because he was actually at the last meeting, but it's maybe something that the Commission could speak to the Interior side on, that walrus, sea otters have benefited from that fund, and expertise from the working group.
So it seems to me, sounds like one of the things we could do is go to Fish and Wildlife when we're in Washington and bring this up as an issue. But it also, we obviously in the past have talked to people on the Hill about when the Prescott program was in trouble, but I don't ever recall talking about appropriated UME money. So it seems to me this is something we could put on our list of things to be talking about on Hill visits.
I'm just going to say this because
oh, sorry. Go ahead, Francis. Since you pointed to me, I was going to just comment that I don't. It's surprising that it was 2005 since there was money appropriated, because somehow magically there's always a little bit in the UME fund, and it seems like there wouldn't be by now, but magically Terry's program keeps them in there. But I will say that a lot of the discussions that we had at the last meeting, we were talking about these sort of repeat UME events or things that are no longer surprising. We kind of know the process that happens, and there's triage going on in the thinking about, well, you know, we're going to have to stop responding or, you know, releasing funds for some of these events that we may still learn some important things about, but they're not as unusual as they once were, and so we're gonna have to focus on the things that are actually really unexpected. And I think that's a shame that we're going to be making those kind of calls. So anything that could be done to sort of top up that already pretty small fund would probably be very useful.
Are you done with that topic?
Yeah.
Okay. So something that I don't think we want to talk about it now. Maybe we don't want to talk about it at all. But if there's a good time to talk about it, it's probably on day 3. But I wanted to flag it now. Well, Alex is here. Is he still here? Yeah. And this comes up in the context of Paul Wade's talk and Robin's talk, and now this issue raised about the impacts of tags on whales in the Arctic. I think there's especially what Alex just said about the improvements in the implantable tags. I think personally that the question of whether the Marine Mammal Commission or anyone else should revisit or ask to revisit the question of tagging North Atlantic right whales should be at least discussed, because I think it's a potentially critical aspect of mitigation and risk reduction to right whales in the current context. And I could go into detail about what I'm thinking, but I just wanted to plant the seed. And maybe at some point someone can brief us on where things stand in terms of understanding the risks of the tagging. As Alex said, there's been improvement, and how has this been evaluated, and is there a new thought on that subject from where it was before?
Anybody want to say anything to that right now, or we just put it on the table for now?
Randy, are you talking about the fully implantable tags or dart tags that are sitting outside the body?
Yeah, I'm talking about fully implantable.
So it's
Did
my
you hear me? Yeah.
it's my understanding right now that no one is authorized to use like the Bruce Mate style type of tag or the tag that Alex uses. And those that have recently been discussed to be used are still the longer variant. And so it's anywhere between 20 and 29 centimeters. And so keeping in mind the letter we just wrote about Brutus whales and how thick the blubber layer is, the Commission's stance would probably be that you shouldn't be using 20 to 29 centimeter tags on North Atlantic right whales based on how thick their blubber layer is. So we can discuss it further, but I just want to flag that for discussion as well. If they make smaller ones, then maybe, but I mean, we've commented in the past about how thick blubber layers are and what type of tags are appropriate and what aren't, so.
If we're in the mood of just highlighting issues to come back to, and since Tom's still in the room, I think we never really pay enough attention to the declining northern fur seal populations, and yet we keep talking about climate change in the Arctic, and we have this huge contrast between what the fur seals are doing up north and what's happening to other pinniped populations. And we also keep sitting there saying we shouldn't leave everything till it's too late, and yet we just keep watching this happen. So I just want to put it on the table as something that we should pay more attention to than we have.
Since we're putting a whole bunch of random topics on the table here, I felt I should bring this up because Catherine Burchak was here and we didn't really have much of a chance to talk about the passive acoustic side. I did want to highlight that one technology that her group uses that has been of enormous benefit has been the use of surplus Navy sonobuoys for the research. She mentioned it briefly. And I wanted to check to make sure this, basically when the Navy has an expiration date on these sonobuoys, they've been donating them. And for something that's free, the amount of use that they've gotten out of them has been tremendous. And I just want to highlight to the Commission that this is a resource that's been tremendously valuable. It's been free. And I'd just like to make sure, and maybe Catherine, if you feel there's ever been an issue getting these things, or there's some risk they may not be provided in the future, maybe you can comment on that. But I think this is something at least to be aware of, and that's been a very useful and beneficial tool, particularly for studying North Pacific right whales. So Catherine's not in agreement. All right. And no problems getting them? You're happy? See? Great.
Actually started in the past several years to actually request proposals to provide brand new ones. So certain groups can get 1 to 2 crates of absolutely brand spanking new ones, which work perfectly. But even the old ones work, 91% of the time, so we're not complaining.
Okay, Doug, go ahead. Make sure you say who you are.
Thank you. Doug DeMaster, Alaska Fisheries Science Center for 4 more days. And then I don't know what happens. Francis's point about the fur seal decline, that is an area that the Commission could probably spend more time on. There's that central western Aleutian corridor. You have fur seals in decline, harbor seals at 50% or less of historic abundance, and you've got stellar sea lions declining at 6 to 8%. There's something wrong with that environment, and it's fur seals going through the passes and stellars eating in the area, and I'm not quite sure what harbor seals are doing, but that Aleutian chain area something's wrong. So I agree with you that fur seals we ought to spend more time on, but it's a bigger issue, I'm guessing, than just fur seals. Thank you.
Well, certainly Alaska and Arctic issues are still a priority in our strategic plan, and while maybe fur seals haven't been front and center, there's no reason we can't be thinking more about that. Okay, I think it's probably a good ending to the day, and I think with that we'll end the session and give one last opportunity if anybody wants to say anything from the table up front here, and if not, we'll see everybody in the morning.